Okay, today let's start the discussion on digestion and absorption of lipids. In our previous class, we have discussed digestion of carbohydrates and proteins as well as their absorption. So, in today's class, we are going to see about these lipids. Now, what are lipids? These are large molecules and they are water insoluble compounds. They are basically are formed of triglycerides. Triglycerides are nothing but the esters of alcohol and fatty acids. So, lipid is an important component of our food. Like carbohydrates and proteins, this will also have to be digested. That means it has to be broken down into smaller component so that it can be absorbed. Now, what are the sources of lipid? Here we have egg, meat, nuts, oil, fish, etc., including milk. Okay, as you can see on the picture there, avocado fruit. Okay, and there are lots of nuts, including our groundnut. Then the vegetable oil. You take any oil that is used. Okay, is a rich source of lipid to the body. Even the meat, any meat, fish or poultry or from any other source, meat is also a source of lipid to the body. Okay. So, now before understanding the digestion of lipid, let us see what is a lipid. As I said, lipid is a triglyceride. It can be either fat or it can be oil. Fat and oil difference is that fat will be solid at room temperature and oil will be liquid at room temperature, isn't it? So, both of these are formed of triglycerides only. Okay, what is a triglyceride? A triglyceride is a ester. Okay, it is a ester and this ester as you can see here, okay, see this is a ester. I think I might be able to use screen picture. Yes, it's here. Okay, it's taking some time. Sorry for that. Anyway, what you are seeing here on the screen is a triglyceride. Okay, and it is basically formed of a alcohol. Okay, here it is a trihydric alcohol because 3 alcoholic groups are present and they are esterified with 3 different fatty acids. Okay, either similar type or different type, 3 fatty acids are present. So, when condensation takes place between these, you are going to see a molecule of a triglyceride. A triglyceride is formed of alcohol, here it is glycerol, and 3 fatty acid chains. Fatty acid is similar to your hydrocarbon that you study in chemistry, formed of carbon and hydrogen, but the difference is that it will also have a carboxylic group. C double bond O, OH group will be present. That is how hydrocarbon is different from a normal fatty acid. Hydrocarbons will be linear chain of carbon and hydrogen, either double or triple bond, but fatty acid will be a linear chain, plus it will also have its one of the end will be will contain carboxylic group. C double bond O, OH group will be present. Such a chain of carbon and hydrogen and oxygen is called as fatty acid. A triglyceride is formed of one alcohol compound, that is a trihydric alcohol here, glycerol, and all the 3 alcoholic group, if they are bound to 3 fatty acids, it is a triglyceride. If 2 of them are bound, then it is a diglyceride. That means you know what is a monoglyceride now, isn't it? Okay, so any lipid, you take either a fat or a oil, basically it is formed of this. Okay, it is formed of alcohol and it is formed of fatty acid. That means during digestion, this ester will have to be broken down. It will have to be broken down into corresponding units like either monoglyceride and 2 fatty acid molecules, or all the 3 fatty acids and a glycerol molecule will be separated. So, that is about the lipid. Now, this lipid digestion, let us say how it takes place. See, when you take the food, you are going to eat. Okay, when you are eating the food goes to the mouth first And in the mouth, you know that it has saliva, and this saliva contains different enzymes. In our one of the classes, we have seen that this has a salivary amylase that helps in digestion of carbohydrates. Okay, alpha amylases are present in it. Similarly, the same saliva also has something called as lipase. Now, these lipases are general class of enzymes that digest lipids. Okay, it is a hydrolyzing enzyme, or it is a hydrolytic enzyme that digests lipid, is called as a lipase. If the lipase is secreted in the mouth, then it is called as lingual lipase. If it is in stomach, gastric lipase. If it is pancreas, pancreatic lipase, and intestine, intestinal lipases. So here we are talking about the mouth. So the lipase here, it's called lingual lipase. Now, what does this do? This lingual lipase actually doesn't help in total digestion of the fat. Instead, it only prepares the fat for further digestion in next part of the digestive tract. So, because of the activity of lingual lipases, it becomes more accessible to the other digestive enzymes. Okay, and here what happens, because of the churning and all, because of mechanical digestion, as well as because of the activity of lingual lipases, the large a globule of the fat will be broken down into small droplets, so that they are ready for further digestion in future. So no much digestion of fat in the mouth. You swallow the food. Okay, food passes through the esophagus and goes to the stomach, and we know that stomach also has gland called as gastric glands. They also secrete different types of enzymes, and here the enzyme that can be used here in our discussion is a gastric lipase, because this is also a lipid digesting enzyme, but it is a weak lipolytic enzyme, so no much digestion of lipid again in the stomach. This enzyme is active at a pH of 4 to 5, and usually it is inactive below 2.5 pH. And we are all familiar that the stomach pH is generally somewhere between 1.2 to 2.5. So anywhere close to 4, that's all, not more not less, not more acidic than that. So here because of this, what happens, gastric lipase will not be much useful, except in a case where you have eaten butter. So you have eaten butter chicken, or you have spread butter on bread and you have eaten, then in that case such butter will be digested in the stomach itself by the help of gastric lipase, and it will be converted into, broken down into fatty acid and the triglyceride. Okay, so that's the digestion of lipid in the stomach. So both in mouth and in stomach, lipid digestion doesn't take place except for the digestion of tributyric acid that is present in the butter. Okay, except that no much digestion of lipid takes place. That means almost all the lipid that you have eaten will be digested in the intestine only. Okay, and this happens because of secretions of liver, intestine and the pancreas. So here the liver plays an important role, because liver secretes salts. So remember, no digestive enzyme is secreted by liver. So it secretes bile salts and bile pigments. Okay, that is a major component of or composition of the bile juice secreted by liver. This bile juice will be then passed to the pancreas, and from there, through a common duct called as hepatopancreatic duct, the secretion of both liver as well as of pancreas will be secreted to the duodenum. So this secretion of the pancreas also contains enzymes that help in digestion of lipid. Okay, and this enzyme is called as pancreatic lipase. In addition to that, it also secretes phospholipase A and B. Okay, then cholesterol esterase etc. are present in the secretion of pancreas. Okay, plus intestine also secretes its own enzyme, and here the enzyme that is used is intestinal lipase. Okay. Coming back to our liver, I said liver doesn't secrete digestive enzyme, instead it secretes bile salts, and these bile salts play a very important role in digestion of the fat, which we will be seeing in next few minutes. Now, what are those salts that are secreted by bile or secreted by liver? So there are actually two major groups of salts. One is thiocolate, the other one is glycocolate. So these are the salts of sodium and potassium, and you can name them as sodium glycocolate and potassium glycocolate, as well as sodium thiocolate and potassium thiocolate. All these salts are the salts formed by the combination of bile acids with the sodium or potassium. Okay, so that means you can name 4 of them: sodium potassium glycocolates and sodium potassium taurocolates. Okay, so these are the salts that play a major role in digestion of lipid, and here by a process called as emulsification. Now what is this emulsification? Emulsification is a process by which large fat globules are broken down into small droplets. Okay, and this droplet will form a milky fluid, and that milky fluid that is formed is called as emulsion, and the process of formation of such a emulsion is called emulsification. I can give you a simple example. Take a glass of water, put few drops of oil in it. What do you see in the beginning? They float on the surface, isn't it? Then thoroughly mix it. When you mix it, when you shake it well, what happens? That large droplet will be converted into small droplets. You can see small droplets in the glass of water. Okay, and the color changes from total transparent to milky, cloudy. Okay, and this is called as emulsion. Okay, and that process of mixing this oil and water is called emulsification. Similarly, in our body also, the food that we have eaten contains lipid. This lipid droplets will be broken down into small droplets by the help of bile salts, and this process of converting large fat globules into small droplets is called emulsification of fat, and the product formed is called emulsified fat. As you can see here on the slide, see, this is a fat globule. Okay, what you are seeing here from here to here. Whereas what happens, these are the bile salts. They bind, okay, to the surface, and they break this large globule into small droplets. You see these droplets, okay, they are broken down into small droplets. Now because of the help of, or because of the activity of the bile salt, larger globule is broken down into small droplets, and now at this stage these lipases are able to act on it. Okay, and they are able to digest the fat, okay, into its components. Now why the emulsification takes place? Emulsification takes place because of the main property of the pancreatic lipases. So these lipases are, they are water soluble. Okay, they are water soluble and they are insoluble in fat. So see, these are water soluble ones. Okay, fat is lipid, so they cannot they cannot penetrate into the fat droplet and cannot digest it. So because of that, they'll be only able to digest the fat on the surfaces. But that fat itself is a large globule, isn't it? So digestion won't take place. It might take hours together to slowly digest surface by surface. To enhance this process of digestion of lipid, it is important that the surfaces are separated. Okay, and that's why what happens, these bile salts will make small droplets. When droplets are smaller, pancreatic lipase will be easily able to act on it. So the main reason why the emulsification takes place is to convert that larger droplet of fat into small droplets. Okay, small droplets, so that pancreatic lipases can act. You see the difference here. See, when this is a large molecule, only on the surfaces pancreatic lipases can act, because they cannot penetrate in, because this is fat, they are water soluble. Whereas when they are broken down into small surface, small droplets, each pancreatic lipase can easily act on these droplets and they can digest it. So that's why emulsification of fat takes place. Okay, so that the digestion can be eased. Now, there are number of enzymes that are present in the pancreatic juice that help in digestion, like pancreatic lipase, cholesterol esterase, phospholipases A and B, phosphodiesterases, phosphomonoesterases, etc. But we'll be focusing mainly on pancreatic lipases and how the activity takes place. Now, recall we have seen the structure of triglyceride in our previous slide. So this is a triglyceride molecule. It is formed of glycerol, okay, and 3 fatty acids. Since there are 3 fatty acids, it is called as triglyceride. Now, we also know that if the digestion should take place, these glycerol molecules should be separated. This pancreatic lipase, it has one more weakness, and the weakness is that this can act on first and the third carbon atom only. Okay, it cannot act on this. So it will act on these 2 points and it will break it. See, here the bond is broken. Here also bond is broken. So this whole triglyceride molecule is broken into 3 pieces. So this is one free fatty acid from here. Okay, this is another free fatty acid that's separated from here. What is left is this glycerol molecule and its fatty acid. See, I said if there is if there are 3 fatty acids linked to a glycerol molecule, it is triglyceride. If 2 of them are present, diglyceride. If there is only one, monoglyceride. So, by the end of the pancreatic activity, lipase activity, what happens? You are going to see one molecule of monoglyceride and 2 fatty acid molecules will be formed. This is because pancreatic lipase can act only on these carbon oxygen bonds, okay, that are present at the end of the or at the ends, not in the middle. So because of that, monoglyceride is going to be formed. Okay, and then it will be anyway digested by some modification, and there is isomerase enzyme which will shift this fatty acid to either any one of these carbons. So because of that, again pancreatic lipase will be able to break them. So this is anyway, that is out of the scope of this discussion. So by the end of pancreatic lipase activity, on a emulsified fat, you are going to see the production of a monoglyceride and 2 molecules of fatty acids will be formed. There are other enzymes also, I said, like cholesterol ester hydrolase, which is also known as cholesterol esterases. So they convert these cholesterol esters into free cholesterol and the fatty acid, just like the pancreatic lipases. And a bit of additional information is that even human milk contains similar cholesterol esterases. Okay, so that's a bit of additional information anyway. Next, there is also phospholipase A enzyme. Okay, this digests the phospholipids. See, because it digests phospholipids, it is called because it digests phospholipids, it is called phospholipase. Okay, and this can digest and convert them into lysolecithin or lysocephalin, depending upon what it acts on. Lecithin and cephalin are phospholipids. So based on what they digest on, lysolecithin or lysocephalin are formed. Okay, phospholipase B will further act on these lysophospholipids and convert them. Okay, separate them. Choline and free fatty acids will be produced out of it. Phospholipase A and phospholipase B. Next enzyme that is also useful in lipid digestion is intestinal lipase. So they also act on triglycerides and convert them into fatty acids. Okay, so here is the summary of lipid digestion. So lipids are acted upon by bile salts, converting them into emulsified fat. So emulsified fat will be then acted upon by pancreatic lipase or intestinal lipases, converting them into monoglycerides and fatty acids. Remember, whenever each molecule of a lipid, a fat or a oil is broken down, you are going to get either one molecule of monoglyceride and 2 fatty acids, or 3 fatty acids and a glycerol molecule is going to be formed. Okay, so this is the digestion of lipid. Now let's see how the absorption of lipid takes place. So absorption of lipid takes place through the again intestinal cells called as enterocytes only, and it actually happens by simple diffusion. Okay, so now when we say simple diffusion, it is not such easy one, because intestinal content contains water, and we know that fatty acids are all they are hydrophobic ends. Okay, so their movement is not easy. So to make their movement easier, so the penetration is easier on the surface of the enterocytes, there will be formation of something called as micelles. So these micelles are simple droplets, okay, that are formed of cholesterol, okay, then the glycerides, fatty acids, and majorly bile salts. Okay, and these are 200 times smaller than the emulsion droplets. We said emulsion droplet. Emulsion droplet is a droplet of the fat itself. Okay, these are much smaller than the emulsion droplets, and these micelles are basically formed of fatty acids. Okay, then there will be some monoglycerides, okay, as well as the cholesterol molecules. They are the components, the small droplets. These they cannot freely move in the watery lumen. So to make their movement easier, so these will be then surrounded by bile salts. They will be providing some kind of support, okay, some kind of covering, okay, and then they will be brought, these fatty acids will be brought to the surface of the enterocytes, and this is for the absorption of the fatty acids and the glycerides, monoglycerides on the surface of the enterocyte. So here again you can see, so dietary fat is broken down by pancreatic lipases. So here, okay, pancreatic lipase breaks them into smaller units, and then they will be covered by bile salt to form the micelles. So the micelles will bring these fatty acids on the surface of the enterocytes. They can be easily absorbed. Please remember, micelles are only to bring these on the surface. They will not be absorbed on the surface of the cell. So what you are seeing here is the alimentary canal, the intestinal here you see this is the intestinal lumen, and these are all the micelles that are formed. Okay, and you can also see the fatty acids and the bile salts that are surrounding it, and these micelles will come on the surface here, and then what happens, the those lipids that are present, the fatty acids that are present, will simply move into the enterocytes. Enterocytes are nothing but the cells of the intestinal wall. So simply they move into the intestinal cells as free fatty acids. Now once they come into the free fatty into the enterocytes, inside the enterocytes they will be then processed, okay, by the help of endoplasmic reticulum. Usually smooth endoplasmic reticulum is involved in it. Smooth endoplasmic reticulum will then form something called as chylomicrons. These chylomicrons are some kind of packages, okay, they are some kind of packages formed of proteins and the fatty acids, where actually these fatty acids will combine with the alcohol glycerol to form triglycerides. Don't get confused. Digestion of triglyceride has taken place in the lumen of the intestine, where they are broken down, okay, and monoglycerides and fatty acids were formed. Because fatty acids and these monoglycerides cannot move freely in watery medium, they were converted, they are transformed into micelles by the help of bile salts. Those micelles were brought on the surface of the enterocytes. They are the cells on the lumen of the cells on the intestinal wall. These micelles will release the fatty acids and the monoglycerides. They can be easily absorbed into the enterocytes. Okay. Once they come into the enterocyte, they will be then processed by the help of smooth endoplasmic reticulum, where these units of fatty acids will reconstitute to form triglycerides. Now these triglycerides will be then, okay, fat or oil, anything, Usually a fat will be surrounded by phospholipid layers, okay, vesicle, which is associated with certain special proteins. Apolipoproteins, they are called special proteins, and the whole structure is called as chylomicron, and this chylomicron will then be released to the circulatory system but not the blood, instead to the lymph. I think we have another slide where it is clear than the previous one. So here is what we see, the absorption of fat or the lipid. I'll just take the different color here. Yes, so the fatty acids and the monoglycerides, see, there is a micelle formation, but the micelle itself will not go inside. Instead, only the those fatty acids will go in. Then they will reconstitute to become triglycerides. By the help of endoplasmic reticulum and Golgi, they are converted into chylomicrons. These chylomicrons will be then passed into the lymph, remember, into the lymph and not to the circulatory system. They are absorbed into the lymph. This is the difference between carbohydrate protein absorption and lipid absorption. Carbohydrate and protein will be added to the blood, but lipids will be added to the lymph. Later on they will be anyway added to the blood. Now what are these chylomicrons? So these chylomicrons are lipoproteins. They are formed of lipids and the proteins. They are the special particles. Okay, and their main function is to transport the lipids in the circulation. See, this is how they are. It is a diagrammatic representation of it. So these are the triglycerides. Okay, these are the triglycerides. Here you see triglycerides, this one, and they are surrounded by these phospholipids as well as packed by special proteins. Okay, because they are formed of lipids and the proteins, they are called as lipoprotein droplets. Lipoprotein particles, you can say, and they will carry this lipid to the lymph. Now, why it goes to the lymph? Why not to the circulatory system? It is because these droplets are much larger. Okay, so that's why they will be taken to the lymph first. As you can see here, those lymphatic vessels are called as lacteals. Okay, they are called as lacteals. So in the villi, after absorption, those lipid molecules will go to the these lymphatic vessels in the form of chylomicrons. Then it will be circulated and it goes to the, it will be released to the circulatory system, to the blood circulatory system at the region of subclavian vein. Subclavian vein will be found just in the shoulder region. Okay, so there is a bone called as clavicle. So in that area, the collarbone, what do you say? So near that there is a vein, and here the lymph will release the chylomicrons, which are actually digested into the constituent units, because there are special enzymes called as lipoprotein lipases. Lipoprotein lipase enzymes will break this lipoprotein into its constituent units. Okay, so then it is utilized by for different purposes, for the production of our own body fat, or for the generation of energy, or as a skeletal unit for the production of different hormones that are needed by our body. For all different purposes, this will be assimilated. Okay, so that is how the digestion and absorption of the lipid takes place. To summarize, the lipid that is taken will not be digested much in the mouth and the stomach. Instead, its digestion takes place in the intestine by the help of pancreatic lipases as well as the bile salts. Bile salts will emulsify the lipid into emulsified fat. Okay, and on this emulsified fat, the pancreatic lipase will act and break it into its constituent units like monoglycerides and fatty acids. These monoglycerides and fatty acids will be packed in the micelles, and then during absorption they will be brought to the surface of the enterocytes. So here only fatty acids and the monoglycerides will be taken in, leaving the rest of the part out. Okay, so these, when they are in the enterocytes, will be again reconstituted to form the triglycerides, and these triglycerides will be then packed by the help of endoplasmic reticulum and the Golgi complex to form particles called as chylomicrons. They are the lipoprotein particle containing the triglycerides. They bring these triglycerides into the lacteals nothing but the lymph vessels, and from there they will be then released to the main circulatory system at the region of subclavian vein. Okay, so that is about the digestion and absorption of lipid in our body. These are my references for today's class. Thank you.